Thank you, Erica. So I'm Ian Clark in Melbourne, and it's my pleasure to chair this session. First of all, I'd like to welcome everybody into the actual webinar today, and to steal a line from Sean Connery, welcome all of my friends who may be tuned in, as well as my enemies. It's a great pleasure to introduce Richard Peat, who is going to talk to us about Kisspeptin neurons and circuits that control ovulation, which is a subject dear to my heart. And just by way of background, I'd like to introduce Richard. He gained his PhD in Bordeaux in 2003. Then he postdoced in Oregon from 2004 to 7, and then at INSERM in Bordeaux from 08 to 10. From there, he moved to the end of the world, Dunedin, New Zealand, where he had a research fellowship in the lab that was established and run by Alan Herberson, and became an independent research fellow there in Dunedin in the neuroendocrine group in 2016, which continued through to 19, and then to become an assistant professor at Kent State, where he's been until the present day. So Richard didn't need to join the Navy to see the world. And I guess for the younger members who are listening in, there's a lesson there that if you want to see the world, you go into a scientific career. So Richard is really fundamentally interested in the neuroendocrine circuits that control GnRH secretion, and particularly Kisspeptin and the associated peptides. His latest sort of area of focus is on the suprachiasmatic nucleus and how that inputs the GnRH system. Just a quick word on his publications. He's got an H factor of 24 with 2,600 citations. He's got some lovely papers in press. One 2015 in the Journal of Neuroscience, and another one 2013 Journal of Neuroscience. These are his first author papers. A third one, in fact, 2011, and a fourth one again in Journal of Neuroscience going back to 2007. And people will recognize that publishing in that journal is no mean feat. There's a PNAS paper back in 2004. So I think we're very fortunate today to have somebody who has worked in a number of labs and has a wide experience in this particular area. And I'd now like to hand over to Richard to present his seminar.
Thank you very much, Ian, for the kind introduction, and I hope you guys can hear me okay. I want to thank the Society for the Study of Reproduction and the organizers of this webinar series to give me this opportunity today to present some of my work. So I'm going to be talking about what we've learned about the circuits that control ovulation in female mice, as Ian said. In the first part of the presentation, I'll be presenting work that we published last year on the role of the central circadian clock in regulating the circuits under physiological conditions. And then in the second part of the talk, I'd like to show you some unpublished observations that we made on these circuits under pathological conditions of fertility. Okay, so it's great to be coming in after last week's webinar by Sue Mentor. As in her wonderful talk, she set the stage for a lot of the things that I'm going to be talking about today. But for those of you that didn't attend, I just wanted to reiterate that fertility is governed by the gonadotropin-releasing hormone neurons, or GnRH neurons, which control the release of the gonadotropin hormones, luteinizing hormone and follicle-stimulating hormone, LH and FSH, and thus gonadal function. So in females, the gonadotropins control the later stages of follicle maturation, as well as ovulation, and also stimulate the production of gonadal steroid hormones, such as estrogen, illustrated here, and also progesterone, which I didn't include in this cartoon. So there's two main patterns of gonadotropin secretion. One is a basal pulsatile pattern of secretion that is found in males and females. And it's thought to be controlled by a population of Kisspeptin neurons in the arcuate nucleus that Sue introduced last week. They're also known as the candy cells. And these cells generate episodes of electrical activity that drive pulsatile GnRH secretion at the median eminence and thereby drive pulsatile gonadotropin secretion. And this is tempered by estrogen negative feedback for most of the ovulatory cycle in females, and also is negatively regulated by testosterone in males. But in females, once per cycle, there is also a surge in LH secretion that triggers ovulation. That's known as the pre-ovulatory surge. And this is brought about by mature follicles secreting more and more estrogen, which eventually exerts positive feedback effects onto the hypothalamus and onto the anterior pituitary. And it's thought that, at least in rodents, this is mediated by another population of Kisspeptin neurons that's located in a preoptic area here in a region that we refer to as the rostral periventricular area of the third ventricle, or RP3V. And these neurons project to the GnRH neurons to relay positive feedback information for the surge. Okay, I just wanted to add to this cartoon that, and I think that Sue talked about this last week, but it doesn't hurt to repeat it, that these patterns of secretion are really important for gonadal function, for fertility, are also modulated by a variety of signals that ultimately inform the GnRH neurons about the internal state of the body. And that could be information about metabolism or a physiological stressor, for example, and about what's going on the outside. And that could be information about time of day, season, or a psychogenic stressor, for example. And these cues are relayed to the GnRH and Kisspeptin neurons via neurotransmitters and neuropeptides that in turn affect the activity of these cells. So before I go any further, I want to show you that there's quite a bit of information that's available nowadays on how RP3V Kisspeptin neurons may mediate estrogen positive feedback. And this is thanks to work that has been carried out over the past 10 to 15 years by a number of labs the world over that has revealed that estrogen feedback activates these neurons in the RP3V. Excuse me. Causes an upregulation of the expression of the Kisspeptin gene, leading to an increase in the production of the Kisspeptin peptide, upregulates a number of ion currents, which in turn increases the excitability of these cells for the surge. And it is likely that all of these changes are mediated through activation of estrogen receptor alpha in these cells. And in fact, the Mentor lab showed recently that knocking down ER alpha specifically or selectively in RP3V Kisspeptin neurons can prevent some of these changes in response to positive feedback, as well as blunt the pre-ovulatory surge. On the other hand, we recently showed when I was in the Herbison lab that driving electrical activity, specifically in RP3V Kisspeptin neurons, results in Kisspeptin release onto the GnRH neurons, which results in an increase in action potential firing in brain slices. So these experiments were carried out using optogenetics. What's illustrated here is that the optogenetic stimulation of RP3V Kisspeptin neuron fibers innervating GnRH neurons cause an increase in firing in these cells. This is sufficient in vivo to cause surge-like increases in LH secretion, okay? So what we have here is a microcircuit that integrates positive feedback in hormonal cues from the ovary, and then relays this to the GnRH neurons for the surge. And this is really important because the GnRH neurons cannot do that on their own because they do not express the appropriate receptors to do that. One of the environmental cues that powerfully controls this circuit is time of day. So there's an abundance of evidence that the surge is controlled by the central circadian clock that's located in the suprachiasmatic. This is clearly seen in rodents, where this has been studied extensively. But I just wanted to point out here that there's also some evidence that this might be the case in some other animal models and potentially in humans as well. But in female lab rodents, which are nocturnal, what's illustrated here is that the surge occurs always, invariably, at the end of the light phase of the light and dark cycle, ensuring that ovulation, which occurs a few hours later, takes place while the female is awake and able to display sexual behavior during estrus, thereby optimizing the chances of fertilization. This is controlled by projections from neurons that are comprised within the SCN to the GnRH neuronal network. And in fact, lesions of the SCN abolish not only the pre-ovulatory surge, but also abolish the estrus cycle altogether. What's been shown over the years by many different groups is that the SCN seems to be relaying daily timing signals to the GnRH neuronal network that will initiate the pre-ovulatory surge, but obviously this is only effective on the pro-estrus stage of the estrus cycle. So there might be embedded within the circuit mechanisms to gate activation of the different players within the brain to trigger the surge only on pro-estrus in response to these daily signals. Some of these projections have been identified over the years as originating in two populations of SCN neurons, which are very important for the generation of circadian rhythms. For example, here the neurons that produce vasoactive intestinal peptide, or VIP, which are located in the core aspect of the SCN, project directly to the GnRH neurons, whereas other neurons that are located in the shell aspect of the suprachiasmatic nucleus and produce the peptide arginine vasopressin, or AVP, these neurons project directly to the RP3V Kisspeptin neurons. And thereby, they may indirectly control the activity of GnRH neurons in an indirect fashion or a polysynaptic pathway. So I just want to point out here that there's a lot of great work that has been done in the past few years looking at VIP projections to the GnRH neurons and their role in triggering the surge, including work by the Boyce lab in the Netherlands, as well as work by the Mentor lab, and work by myself when I worked in the Herbison lab that showed that VIP can stimulate the activity of GnRH neurons. And so that circuit may play a substantial role in triggering the surge. But today I want to focus on the projections from SCN vasopressin neurons to the Kisspeptin neurons. And that's because this is a part of the circuit where potentially circadian inflammation can be integrated with hormonal feedback. And this, as I said earlier, cannot be achieved by the GnRH neurons because they do not express the appropriate receptors. In addition to that, elegant work carried out in the De La Iglesia laboratory in Seattle showed that the part of the SCN that contains the vasopressin neurons comprises an oscillator that is involved in timing Kisspeptin neuron activation and timing the LH surge later on. So we think that this part of the circuit is very important in triggering the surge. To be fair, we're not the first ones to think that this pathway is important. And in fact, there's a lot of evidence that has accumulated over the years that it might play a role in the LH surge. And I'm showing you some examples here. First of all, here, the vasopressin V1 receptor antagonist administered to foresters rats causes a blunting of the pre-ovulatory surge. Although to be fair here, other studies have not been able to reproduce that. On the other hand, the RP3V, and what's illustrated here is the interventral periventricular nucleus of the RP3V. This area is full of vasopressin V1 receptors, in particular the V1A subtype of vasopressin receptors. And it was shown by a couple of groups, including the Kriegfeld lab in California, as well as the Liposits lab in Hungary, that Kisspeptin neurons in the RP3V receive innervation by vasopressin immunoreactive fibers. And lastly, when I was in the Herberson lab, we observed that exogenous vasopressin applied to brain slices can increase action potential firing in RP3V Kisspeptin neurons, as illustrated here. So we have many different pieces of the puzzle, and what we wanted to do here is, reconstitute that full image and get a full picture of how this circuit might be controlling RP3V Kisspeptin neuron activity. And so we hypothesized that activity in these SCN vasopressin neuron and their projections would drive Kisspeptin neuron firing via the release of vasopressin. On the other hand, as I said earlier, the SCN is sending daily signals to the GnRH neuronal network. The surge only occurs once per ovulatory cycle. And therefore, there might be some sort of gating mechanism, and we hypothesized that the output of this circuit would vary during the oestrus cycle, thereby contributing to gating the surge to only pro-oestrus. This was the work of Bradley Jamieson, who's my former PhD student. He's now a postdoc at the Francis Crick Institute in London, UK. And he looked into these questions using optogenetics and brain slice electrophysiology. And so we had mice that express the DNA recombining enzyme pre-recombinase, specifically in vasopressin neurons, which enabled us to express the blue light activated cation channel rhodopsin, which is illustrated here, specifically in the SCN vasopressin neurons using serotoxic surgery and serotoxic injections of viral vectors that carried a pre-dependent sequence for channel rhodopsin and cherry. And so after transfection of SCN vasopressin neurons, using blue light to activate the channel, which causes an entrance of positive charges inside the cell, thereby depolarizing the membrane potential and causing action potential firing, we could potentially drive the activity of, selectively drive the activity of SCN vasopressin neurons. And we've done some control experiments showing that indeed, when we record from single SCN or single transfected SCN vasopressin neurons, we can readily drive their action potential firing using short pulses of blue light. So these mice also expressed a green fluorescent protein, specifically in Kisspeptin neurons. So this is a mouse strain that was developed by the Elias group in Michigan. And this enabled us to essentially cut brain slices that would include the RP3V. And these slices would contain GFP expressing Kisspeptin neurons that we could then target and see for electrophysiological recordings. as well as the cut-up axons of SCN or channel rhodopsin expressing SCN vasopressin neurons that project to this area. And you'll see later in the talk that we have evidence that these projections are indeed within the RP3. And so this way, Bradley was able to, using blue light, selectively activate these axons to cause them to release their contents and see the effect of that stimulation on the electrical activity of single Kisspeptin neurons using electrophysiology. So in the next few slides, I'm going to show you a number of different electrophysiological traces. And most of these will be traces obtained from what we call loose patch recordings. So this is a type of cell-attached recording, cell-attached patch clamp recording, I should say. And it's a form of targeted extracellular recording that enables us to monitor the action potential firing of single neurons, in this case, the Kisspeptin neurons, without disturbing their internal milieu. So in the traces that I will show you today, you will see, and in fact, you've seen that in a couple of slides that I've shown you already, you'll see these downward deflections, which may or may not be patterned depending on the example. And each of these deflections correspond to a single action potential as illustrated here. Okay, so let's see now what the effect of activating these fibers is on the Kisspeptin neurons. So Bradley started with slices obtained from pro-ostrus mice. And on that day of the cycle, which is when the surge is generated, he saw that activating channel rhodopsin expressing axons coming from the SCN resulted in a slow onset and sustained increase in firing in about 60% of the Kisspeptin neurons that are recorded from. So the kinetics of this response, which has a delayed onset and a sustained increase in firing that outlasts the stimulation, was reminiscent of what a neuropeptide might be doing, acting at a G protein coupled receptor. And indeed, Bradley was able to show that using the mannan compound, which is a V1 vasopressin receptor antagonist, he was able to prevent this increase in firing in pro-ostrus Kisspeptin neuron. So this is all quantified here. You can see that in about 60% of the cells, there was an increase in firing after the stimulation. This increased the firing overall significantly, and this was prevented in the presence of the mannan compound. In fact, this was significantly different between the two groups. Bradley then looked at slices from diastrus mice. So that's the stage of the cycle that precedes the generation of the pre-ovulatory surge. And although he could find a number of cells that did display a similar behavior with a delayed increase in firing, overall, as a population, Kisspeptin neurons on diastrus did not show an increase in firing. And in fact, the effect of activation of the SCN fibers was significantly different, significantly smaller than what it was in pro-ostrus. So right here, you can sort of see that depending on the stage of the estrous cycle, the output of this circuit might be different and potentially optimized to activate the Kisspeptin neurons at the time of the surge. And this was confirmed by looking at slices from ostrus animals in which, as shown here, there was no delayed increase in firing in Kisspeptin neurons. But as you can probably see on this trace here, the situation was completely different to what we saw in the other cycle stages. Excuse me. And so what we saw was a decrease or a suppression of action potential firing in response to the stimulation. And this was seen, so this is here the response during the stimulation this time. We saw this suppression in about 75% of the Kisspeptin neurons on ostrus. We only saw that in a couple of other cells in diastrus or pro-ostrus out of 40-something recordings. And in fact, we think that it is specific to oestrus because even in the presence of the mining compound, which abolishes this increase in firing, we could not see a suppression of firing during the stimulation. So we do think that it was not masked by vasopressin-dependent excitation, but rather it is something specific happening on oestrus. I don't want to spend too long on this, but Bradley did a lot of work to try and figure out what were the mechanisms for these changes in responses. First, he looked into why there was an absence in delayed activation in oestrus Kisspeptin neurons, and he was able to show that there was some sort of downregulation of V1 receptor signaling in oestrus Kisspeptin neurons, and he showed this by observing that exogenous vasopressin was unable to activate oestrus Kisspeptin neurons, whereas, as expected, it would trigger firing in proestrus or in diöstrus. During the stimulation, and again, I'm not going to show you the details of the data in the interest of time, but Bradley found that the suppression of firing was dependent on GABA release, which most SCN neurons co-express, including the SCN vasopressin neurons, acting at GABA A and GABA B receptors. But what was surprising to us was that we found very little evidence of direct synaptic transmission between SCN vasopressin neurons and RP3V Kisspeptin neurons. So this GABA-dependent suppression of firing seen on oestrus during the stimulation must be mediated via an extrasynaptic or a polysynaptic mechanism. And we, at this stage, we don't have a good grasp on that. But regardless of the mechanism, to summarize all this, I showed you here that, and I hope I convinced you that SCN vasopressin neurons indeed can regulate the activity of RP3V Kisspeptin neurons, and it would appear that they do so by releasing vasopressin, and it would seem that at the transition between diöstrus and proestrus, there's I guess an upregulation of the output, the vasopressin-dependent output of this circuit. But surprisingly, after ovulation, there is a dramatic shift in the output of this circuit, such that vasopressin-mediated excitation is suppressed completely on oestrus, and the output instead shifts to a suppression of firing that is mediated by GABA on oestrus. So, as I said earlier, the SCN is sending daily signals to Kisspeptin neurons and to GnRH neurons, and we think that this buildup in excitation between diöstrus and proestrus on the one hand, and then the shift in the output of the circuit after ovulation on the other hand, might be playing a role in preventing inappropriate surge generation. For example, after ovulation, the one thing you want to prevent is that another surge in LH secretion be initiated ovulation. So another thing that's apparent here on this schematic is that, as you all may know, gonadal steroid hormones greatly fluctuate throughout the oestrus cycle, while these changing in the output of the circuit might be going on. And we think that possibly these fluctuations might have an influence on how these shifts in the output might be brought about. So the data that I showed you so far is consistent with activity in SCN vasopressin neurons, stimulating proestrus Kisspeptin neurons, thereby providing potentially a drive on the GnRH neuron activity in preparation for the surge, and thereby for ovulation. So in the second part of this talk, I'd like to show you some evidence that this circuit here might be sensitive to hormonal dysregulation occurring during pathological conditions of fertility. And in particular, we became interested in the impact that polycystic ovary syndrome, PCOS, may have on this circuit. So as a lot of you may know, PCOS is a prevalent endocrine, or neuroendocrine, I should say, disorder that is associated with elevated testosterone secretion, polycystic appearance of the ovaries, and ovulatory dysfunction. And clinically, the presence of 2 out of these 3 core symptoms is considered the criterion for diagnosing PCOS. The causes of PCOS are currently unknown, but it's likely that it's the result of a combination of multiple factors, including genetic factors, metabolic dysfunction or dysregulation, as well as potentially elevated exposure to elevated androgen hormones during the perinatal period. And so, using early exposure to androgen hormones, many labs have developed animal models that replicate some or all of PCOS symptoms. Work in these models have revealed great disruptions in negative feedback regulation of the GnRH neurons, increased LH secretion, as well as neuroendocrine alterations in the circuits that control GnRH secretion and LH secretion. A hard time switching slides here. In fact, there was evidence from prior work by Ray Silver's lab in 2008 showing that females or female rats that were ovarectomized had a certain level of androgen receptor expression in the suprachiasmatic nucleus. But interestingly, the expression of the androgen receptor was significantly and dramatically increased by treatment with testosterone. So we hypothesized that in addition to the neuroendocrine disruptions that I've listed before, the increase in testosterone secretion that's seen in PCOS animal models and potentially in PCOS patients might dysregulate the circuit that I told you about before between SCN vasopressin neurons and Kisspeptin neurons, and that this might have an influence on the regulation of the preovulatory surge. So to address this question, we used a well-characterized prenatal androgen model of PCOS in our mice. So this is a model that has been used in many species, and was pioneered in rodents by the Mentor Lab in the early 2000s and has since been adopted by many other labs. This model, we treat pregnant females, with injections of dihydrotestosterone at the end of pregnancy. So dihydrotestosterone is an androgen that signals through the androgen receptor and cannot be broken down into estradiol. So we treat these animals with dihydrotestosterone or with the oil vehicle as a control on gestational days 16, 17, and 18. The offspring of these mothers that have been thus exposed to androgens in utero display as adults excess androgen secretion, elevated LH secretion with increased pulsatile LH secretion, as seen in many patients with PCOS, disrupted estrous cycles with animals sitting mostly in diestrus, sometimes in estrus, but never or very rarely in proestrus, and decreased ovulatory function. So we decided to use this model to see if there was any alteration in circuits that we studied before that we showed controlled Kisspeptin neuron activation at the time of the surge. So for the remaining of the talk, I'll be referring to these prenatally androgen-treated animals as P and A, whereas the oil-treated vehicle controls will be referred to as the P and V animals. The first thing that we asked was whether indeed in female mice there was expression of the androgen receptor in the SCN. and in particular in SCN vasopressin neurons, and what the effect of PNA would be on that. And so for this, we collaborated with a group of Alicia Moore, Nika Cullen, and Mike Lehman here at State University in the Brain Health Research Institute, because this group had recently published a very nice paper where they looked at changes in gene expression in the Candy neurons in PNA mice using multiplex in situ hybridization. So we thought that this approach would enable us to answer the question. And so Alicia and her technician Daylor carried out RNA-scope in the SCN of PNV and PNA mice. And what I'm showing you here on the left is the expression of the vasopressin gene within the SCN. You can see expression in the shell aspect of the SCN as expected. And as is quantified here, you can see that PNA didn't have any substantial effect on the expression of that gene in the SCN. But interestingly, Alicia found that there was expression of the androgen receptor mRNA within the SCN. This is found in the shell, but perhaps more within the core aspect of the SCN. And as I hope you can appreciate here, there was an upregulation in the expression of the androgen receptor messenger RNA in PNA mice. And so Alicia then looked specifically at the cells that express the androgen receptor in the SCN. And I just want to mention here that if you're a little colorblind like me, you must be having a great time looking at this, this figure. But nevertheless, as I hope you can see here, using DAPI staining to locate individual cells in the SCN, Alicia was able to show that individual SCN cells, or the number, of individual cells within the SCN that express the androgen receptor mRNA increased in PNA mice, as is quantified here, and this turned out to be significant. Interestingly, she also found that a proportion of vasopressin expressing SCN neurons in the SCN also express the androgen receptor. So this is illustrated here with these filled arrowheads in PNV and PNA mice. But somewhat disappointingly for me, but interestingly, she found that although there was a trend for an increase in PNA, the proportion of vasopressin cells, or the number of vasopressin cells that also increased the androgen receptor mRNA did not significantly increase with PNA. And so it would appear that the increase in the number of cells that express androgen receptor in PNA is accounted for by cells that do not express the vasopressin gene, as is illustrated here. So this showed us that SCN cells, including SCN vasopressin neurons, may be sensitive to circulating testosterone because they express the messenger RNA for the androgen receptor. And in fact, PNA, which is associated with elevated testosterone, dysregulates the sensitivity of the SCN to testosterone. The next question was, are the projections from SCN vasopressin neurons to the LP3V Kisspeptin neurons altered in any ways in PNA mice? And so this is something that we did in collaboration with Rebecca Campbell back in New Zealand, using track tracing. And in these experiments, Bradley used a similar approach where he injected, using stereotoxic surgery, viral vectors that carried a Cre-dependent sequence for mCherry in the SCN of vasopressin Cre mice. And then using immunochemistry for mCherry and Kisspeptin, he could then trace the projections of transfecting neurons and determine the interactions of these projection fibers with Kisspeptin neurons within the LP3V. So Bradley found that, as expected, in the LP3V of PNV animals, fibers, mCherry immunoreactive fibers originating in the SCN do reach the LP3V. But as I hope you can appreciate from these pictures here, in PNA animals, these seem to be much less. And in fact, quantification of this, of the density of these mCherry immunoreactive fibers within the RP3D revealed that there was a significant 45% reduction in the integration of the RP3D in PNA mice. And this couldn't be accounted for by differences in the amount of transaction within the SCN, as illustrated here, because we didn't see any changes in the two groups. These fibers, these anterior immunoreactive fibers, interacted with a proportion of Kisspeptin neurons in the RP3D. So here is shown the RP3D of a PNV mouse that shows that about 45% of Kisspeptin neurons receive close oppositions, as illustrated here, by anterior immunoreactive fibers. You can see here, for example, or here. Interestingly, I forgot to delete this slide, my apologies. Interestingly, in PNA mice, despite a constant number of Kisspeptin neurons or Kisspeptin immunoreactive neurons in the RP3D, he found that there was a decreased proportion of RP3D Kisspeptin neurons innervated by anterior immunoreactive fiber. And please ignore that 45% reduction. In fact, it's more like a 30% reduction. But nevertheless, there was a significant decrease in the number or the proportion of Kisspeptin neurons receiving close oppositions by anterior fibers. And consequently, there was a significant decrease in the number of the average number of close oppositions per Kisspeptin neurons in the RP3D of PNA mice. So this revealed that potentially the direct innervation of Kisspeptin neurons by SCN vasopressin neurons was decreased by elevated testosterone in PNA mice. Skip over that real quick. So the natural next question was to investigate whether PNA had an influence on the function of that circuit. And so this became quickly very frustrating for Bradley mainly, and for me as well, because as I said earlier, the PNA mice are mostly in diestrus, sometimes in estrus, but never found in proestrus. And proestrus is the stage of the cycle where I told you that the output of this circuit is the most prominent and the most able to increase activity in Kisspeptin neurons. So we were left with comparing diestrus PNV mice with diestrus PNA mice. And perhaps not surprisingly, as is illustrated here, Bradley found that PNA did not alter the function of the circuit at baseline in diestrus. So he wanted to dig a little deeper into that and looked at whether Kisspeptin neurons in PNA would be more or less responsive to vasopressin. And so he performed some experiments with exogenous vasopressin, but he found that PNA did not impair the responses of Kisspeptin neurons. So it would appear that, at least at baseline, the circuit is perfectly functional despite the decrease in the integration. Okay, so to summarize all this, I hope that I convinced you today that there's a projection from SCN vasopressin neurons to the Kisspeptin neurons that can drive the activity or can activate the Kisspeptin neurons in the RP3D, which potentially drives the entire circuit to cause activation of GnRH neurons for the pre-ovulatory LH surge and thereby regulating ovulation. And also showed you that cells within the SCN do express the androgen receptor, including some vasopressin SCN neurons. Although I drew it here as the ones that project to the RP3D, we actually don't know which cells express the androgen receptor, whether or not they project to the RP3D Kisspeptin neurons. But nevertheless, we think that this is really important because under conditions of disrupted fertility and in response to increased testosterone secretion induced by prenatal androgen treatment in females, there is somehow somewhat of a dysregulated androgen receptor mediated signaling within the SCN. that disrupts the projections of vasopressin SCN cells to the LP3V Kisspeptin neurons. And that, along with impaired negative feedback regulation of GnRH neurons and with disrupted estrous cycle, we think this prevents the estrous cycle dependent plasticity in the output of this circuit that would normally take place, thereby dysregulating the control of Kisspeptin neurons activation, and thereby not driving the GnRH neurons and dysregulating the pre-ovulatory surge potentially, and potentially contributing to impaired ovulatory function in PNA mice. All right, so that's all I had for today, and I will stop here. I want to thank the members of my lab here at Kansas State University, as well as my lab members back in the day at the University of Otago in New Zealand, obviously including Bradley Jamison, who did a lot of this work, but also Greg Bauer and Simon Thomas, who were providing a lot of technical help with this project. I want to acknowledge my collaborator at the University of Otago, Dr. Rebecca Campbell, as well as my dear collaborators here at Kansas State University and the Brain Health Research Institute, Dr. Alicia Moore, Dr. Colden, and Dr. Lehman. And I want to thank you for your attention, and I'll be happy to take any questions.
Thank you very much, Richard. Excellent talk.
Thank you.
Very much focused on a particular issue that is still, I guess, at the frontier. That is how the SCN is functioning to control the diurnal rhythm in the rodent. So I'm looking for questions and answers, and I noticed that some people have put their questions into chats. So Sumenta is first up, and she is interested in the androgen receptor data in the SCN and wonders whether you've considered using a surge induction model in the PNA animals to look at the effect of estradiol on the circuit and how that interacts with the PNA model.
Yeah, thanks for that question, Sue. That's a really good question. And so I'm going to give you a little bit of background. So Alicia Moore showed several years ago that, in fact, it would appear that the PNA treatment doesn't seem to reprogram the circuits that control the pre-ovulatory surge. And this comes from experiments where she ovarectomized PNA animals and used a estrogen induction of the surge, and she found that it was perfectly normal compared to the PNV animals, which suggests to me that potentially what's going on here is that instead of a reprogrammation of the circuits, which does occur in, for other aspects of the effects of PNA on these neuroendocrine circuits, instead of that, we have a diseased ovary, so to speak, that secrete testosterone during adulthood, thereby disrupting the function and the projections of these circuits, and thereby also disrupting the estrous cycle, which thus prevents the circuit to express its proper plasticity, functional plasticity at the time of the surge. So the short answer is no, we haven't looked at that for that very reason, because we think that potentially we wouldn't find anything I guess that would be helpful to us or relevant to that particular question. One possibility would be to use a surge model that is treated or not with testosterone to try and get at that particular question to see if the SCN vasopressin to LP3V Kisspeptin neuron circuit is disrupted then.
Okay, so the next question is from Daniel Bernard, who thanks you for your great presentation and suggests that if he's remembering correctly, and I'm sure he does, that clock mutants and B-mal knockouts still ovulate. So is there still a daily AVP signal from the SCN to the Kiss neurons in those animals? And how can they still ovulate or still surge if the daily signal is lost?
Yeah, so that's also an excellent question. And this is a very puzzling observation. So indeed, sorry, I'm just going to turn off my email real quick. I just had a notification. So indeed, in clock mutants or B-mal mutants, there is still ovulation, and these animals are able to have pups. However, they, as far as people have been able to determine that, they do not mount a surge, which is even more puzzling because it suggests that they're able to ovulate without the neuroendocrine control provided by the LH surge. So this might be a case where the surge is either mistimed or of a very small amplitude, which is still, I guess, sufficient to drive ovulation, but wouldn't be detectable using conventional hormone detection assays and so on and so forth. So I guess I don't have a clear answer to that. I don't know. My thinking would be that the daily signal would be severely disrupted in clock and B-mal1 mutants to the GnRH neuronal network. The state of the circuit between SCN and the GnRH neuronal network is unknown as far as I'm concerned. But I would guess that it would be somewhat disrupted.
So Jennifer Hill also indicates that you gave us an excellent talk, Richard. So, and she wants to know whether you've examined the acute effects of androgens on AVP SCN neurons.
No, we haven't looked at that at all. And so this is an interesting direction, definitely, since there's expression of the androgen receptor in there, and that upregulates in response to PNA. It would be very interesting to see if there's anything that we can pull from either using electrophysiology or any other, I guess, any other marker of cell activity, if we could pull any effects of acute testosterone on these cells. But that's something that we haven't gotten into yet.
Yeah, so if I could just take the liberty of asking a question here, Richard. You mentioned that at one stage in your talk that percentages of neurons gave you responses to firing responses to optogenetics around about 50%. And I recollect that there is only around 50% of the AVPV Kiss neurons that express the estrogen receptor. And you also only have a percentage gain with the AVP input to the Kisspeptin neurons. So how do you see that? I mean, what are these other Kisspeptin neurons doing?
Yeah, so that's a great question. So the short answer is I really don't know, but I can speculate on a couple of things. So Louis Boehm showed several years ago that using a genetically encoded transsynaptic tracer, they were able to show that only about 35% of the Kisspeptin neurons in the RP3B project to the GnRH neurons, despite the fact that most GnRH neurons will receive an input from these cells. And it would appear that most of the cells that were projecting to the GnRH neurons expressed estrogen receptor alpha. So I would speculate that potentially those Kisspeptin neurons that receive input from the SCN would also, or would overlap with the ones that project to the GnRH neurons. At least that would be my dream situation. Now, for the other Kisspeptin neurons that are within the LP3D and not projecting to the GnRH neurons or potentially not receiving an input from the SCN, can only speculate. So we know that potentially these neurons project to the other population of Kisspeptin neurons in the arcuate nucleus. They may establish reciprocal connections within the RP3D, and they may also project to other hypothalamic areas. For example, the group of Julie Becker years ago showed that Kisspeptin neurons are involved in regulating sexual behavior in mice through projections to the ventromedial hypothalamus. So it could be that there's, I guess, subsets of Kisspeptin neurons that fulfill different aspects of the neuroendocrine control of reproduction, with ones driving the LH surge, whereas others may contribute to coordinating this with sexual behavior, whereas others may receive input from the circadian clock to make sure that everything happens in a timely fashion.
Good. So I'll go to Joan Jorgensen, who asks you how you can apply your findings to primates in light of the fact that A. Terasawa has shown that the GnRH LH surge is independent of time of day in primates.
Yeah, so definitely there's many animal models that do not show timing of the surge. This has been clearly established. So I would say that I guess in those animals that lack this timing, it's difficult to envision a role for these projections if they do exist in regulating the LH surge. But I want to mention here that, and I think I alluded to that there's actually evidence in humans that the LH surge is timed, unlike the situation in primates, in other primates, I should say. In that, I'm thinking of one paper in particular that showed back in 2002 that the initiation of the LH surge in women is time locked to the daily cortisol rhythm, indicating, and the daily cortisol rhythm, as many of you may know, is controlled by the circadian clock. Therefore, this suggests that the initiation of the surge might be under the control of the circadian clock as well in women. So I think for humans, I think this we know too little at this stage for the control of the LH surge by the circadian clock. But I would say, to directly answer the question, in primates, this circuit is probably of little relevance, and that probably goes to for other species that do not show a time surge.
Fine. In view of the time, we'll just take this one last question from Catherine Christian Hinman. And she turns your focus to the VIP-containing neurons in the SCN and asks whether the effects of androgens on the VIP neurons are known, and how does that affect VIP transmission from the SCN to the GnRH neurons, or in fact, GABA transmission for that matter.
Yeah, so that's a very good question. I off the top of my head, I don't think this is known, but I may be wrong. This is definitely an area that that would be great to get into. Like I said earlier, there's pretty good evidence that SCN projections, SCN VIP projections to the GnRH neurons play a role in the surge, and so. As I showed you, there's a lot of androgen receptor mRNA expression in the core aspect of the SCN, and that's where the VIP neurons are found. So it is possible that that these cells express the androgen receptor, and as such, they might be sensitive to fluctuations in testosterone in PNA mice, and who knows, that might dysregulate the projections to GnRH neurons.
Well, thank you very much, Richard. I'm afraid our time is up, and we're very grateful to you for what you presented today. And I also thank the participants who have been with us today. I think it's been a wonderful session. So goodbye.
Thank you very much. It was a pleasure to be here. Thank you very much, guys.